My screen is visible. Okay. So, see, we have to take this not as a as a story, not as something that you just learn and take it as a story. There will be a lot of there I have spoken lot of methods. I've told about different ways. You have to also learn, and that is why in my initial slide, if you remember, I gave this roadmap. So all of you, I think your take home message would be you have to have this roadmap, and it should be very clear in your mind that from where you are starting and from where you are ending and what your exact objective is. Once you have this, see, there will be roadblocks, like phase problem was one roadblock, and how we tried to solve or how we tried to remove this roadblock to get this phase information out of our X-ray diffracted patterns. And then once we had done that, what is the preliminary step we did in getting a structure, how robust is our structure, how better I can refine it, and then come out with a structural elucidation which is as good as the real one, right? So this should be as clear like a roadmap. So why I'm telling this repeatedly, a small example, like when you start from your house to this college, to the university, to your class, okay, you know exactly what are the different ways by which I can go, right? There may be multiple ways, and at like peak office time there may be some traffic. So okay, I'm going in this time, this is the best method, this is the best route I can take so that I reach my destination perfectly and well ahead in time, right? And once you know the road, you are in your destination. But suppose you don't have a roadmap, then what happens is you take a step, then you ask, you take feedbacks, and then you come to know, oh, I should have taken the previous left turn, or I should have taken that, from that circle I would have gone to the right side. So something like that. So you do lot of errors and come back and again redo it, come back again redo it, and finally you reach to a destination if you have that determination. That is true, but in that process you waste a lot of time. Now why I'm telling this, because people who have initially started to identify the structure of proteins, they have done this method. Nowadays you have very high end softwares are there, you have very well designed algorithms are there, your number of, as I showed you, you have lot of data in the form of a library which is available. So all you have to do is you follow the steps. It should be clear in your mind what you want to do it, and then definitely within a time frame you will be there in your final structure. Okay, so moving ahead, I'll just come to this slide. Yeah, now as I said, you have multiple ways or multiple roads to reach to your destination, but which model will best suit for my sample? That is where a big question mark applies. Okay, the first question is whether this model is suitable for my sample. Okay, if you say okay, this model is suitable, then the question is why it is suitable. You have to find out some parameters why it is suitable, because this is done on similar type of samples. Suppose a similar type of protein has already been done and we have found that they have deciphered with this method. That is one can be. Or you've referred to some papers, some publications where they have followed this method for a protein, and that protein is very similar. You believe it is very similar because the structure or the amino acid side chains, or you have the different from that paper you find out that structure may be my native protein structure will be similar to that. So all these informations you have at the back of your mind, and you should know and you should plan your pathway that way, right? So what is the best method? Now my suggestion is, or anybody working in crystallography will always tell you, don't use search models blindly. If you do blindly, then it will happen, you will go one street ahead and then somebody will tell you, no, you should have taken the previous left turn. So you will do a hit and trial, errors will be more frequent, and then your time taken will be more, right? So first is, higher the sequence identity, the easier the molecular replacement. As I said, 20% is minimum. You should have at least minimum 20% molecular identification done with your native protein and with the structure you are trying to find out in the library. But it's always better to have a higher. The higher the better. The higher the sequence identity, the easier is to do the molecular replacement methods with your native protein. Okay. So first is higher. Then second is, if there are particular regions, okay, so particular regions of low sequence identity, trim side chains, or like alanine, glycine. So why I'm telling this, suppose it is not easy for you to work out on the exact structure of your native protein, and you are not you are not able to get it in the library so try simple, try with the a low sequence identity, so like the side chains. So if I have distinct side chains in my native protein, try it. Like suppose I showed you that information with the image of a hand, right? So in this hand is your protein, and you are not able to get the entire structure of your hand. So you start with one finger. I have one finger, and try to find this finger is unique to my protein structure, and I'll try to start my identification with one finger, then go to two fingers, then multiple fingers, and then try to get rest of it. So things will be in that way much easier to come to the what model you are trying to do it. Then moving ahead, if a domain in the probe does not exist in the target, delete this from the search model. This is very, very important. Suppose you are trying to find out the hand, and this is the hand of a human being, but in your data you do not have, for example, suppose you do not have the structure or a picture of a human being. You have all the animals, you don't have a human being. Then no matter how much you try to find out with the sequence identity, how much you try to find out with the single side chains, right, you will not be able to find the domain, because that domain, this particular domain, or the main structure of your protein does not exist in the library. So in that case, delete it from the search model and try to find out in different search models. So this model is not going to work for you when your domain in the probe does not exist. Okay, so you have to go to a different search models where this domain exists, and then only you can. Now if the domain in the probe is flexible, okay, now why I'm saying this is very, you have to understand it. If it is flexible, delete this from the search model, or better, use the two domain as separate search models in the same molecular replacement protocols. Now what does this mean? This means, suppose I have a domain, and this domain conformation or structure is not fixed, it keeps changing, it is flexible. Now if I use one particular type of model, I may get the structure, and there are also chances that if I use another type of approach or search model, I may also, this domain may also suit for that. So not to take a chance, try with all the search models which and which will be suitable for this type of domain, which is flexible, because out of these two, either of one of this model, it will suit better, and that model you have to take and move ahead, right? So when you are trying to figure out the MR protocol, molecular replacement protocol, in that case you have to see if your domain in the probe exists or not. If that domain in the probe doesn't exist, then delete it. Don't even try of that search model. Now it can be your domain in the probe can be flexible, so it is also possible that you might have to use two or more search models which will be suitable for your domain, right? So depending on if your domain is flexible, you have to also have an idea for the same. Right. Now, there are several models, yes, as you saw, and you have to combine them also. So at times, using just one model may not help it. Okay. So just one model may not be useful. You might have to, or you might need to combine multiple of these molecular replacements and using an ensembling phasor. So ensembling phasor is just is another method, or a phasor is basically a technique where you ensemble or you put multiple MR techniques, okay, MR methods or models into your sample, and then from there you finally get the best model which can be used and you get the structure, right? So how you are going to do it? First of all, the higher sequence of identity, easier to do the molecular replacement methods or models. If you're not able to find, okay, start with the side chains where alanine glycine is high, particularly it will be easy. So start with side chains. If your domain does not exist, domain in the probe does not exist, forget about that model. If your domain in the probe is flexible, try with multiple MR methods. Which model fits best, you have to see. At the end, even if with multiple models, also you need to use a phasor to ensemble all the methods together and come up with a particular model, right? Now, what is a phasor? Well, I've been talking about, right? So phasor is basically likelihood. Now likelihood is, suppose I'm an individual, there is another person who looks very much similar to me, right? And when we stand together, what is the likelihood, okay, what is the likelihood percentage of him looking like me? So that if he is walking or if he is moving out from my office, from my apartment, people will take him as me walking out from my office. Okay, so if the likelihood is very high, people will think that this is the same person, and that is what is likelihood. Now, likelihood in molecular replacements is like the Patterson based molecular replacement methods assume there are no errors in the data. So we are talking of Patterson method because from there we have the angular information, and clearly this information, once we have done it like from the Patterson method, if we say there are no errors in your data, this is a wrong statement. There cannot be, because it is entirely based on a predictive model. So when you are predictive model, you know partially some information, like you had the electron density. From the electron density, okay, based on the intensity in the X-ray diffraction patterns, you got the electron density. Based on the pattern of the electron density, you got the structural information. Based on the phase by different methods, you got the angles and everything, and then you are predicting a model, right? Now this model is a prediction, it is not 100%. You have informations which you need basically to predict a structure. Once you have collected that information, now you tell the software, you tell your computer, okay, I have done my bit, I give you this information. Now based on this information, give me a structure which is stable enough and which will give me F factor close to this or approximately this and a phase value of this. So these are the parameters which you gave. Now the model or the software will try to find either from the library or as a de novo, it will try to give different structure and try to give you the best fit model. And it will also calculate, once a model it has given you, it will also give you the F value and it will give the phase value. Now once this F value and phase values are very close to the previous, then you say this model is fitting into, or this is my sample structure, native protein structure, right? Now again, this is not 100% error free, because there will be some assumptions, because we have done certain part based on assumptions only. These are all predictions we are doing it, and that is why the likelihood ratio or the likelihood percentage is very important. Now likelihood based methods explicitly are model errors, right? So what is the maximum likelihood? You have to understand that, because based on that, how much your data, what you have, or the model which you have identified, is actually representing or close to the actual structure of the protein which you are studying. Clear? So maximum likelihood, how you're going to do is, the best model is the one that is most consistent with the data. Okay, so what is this data? The f value, the phase value, all this data you have to see. So you have come up with a model and you try to change with different rotations, translations, and then with every rotation and translation angles or movements, you try to find the f value, structures, functions, structure functions, and you want to do the phasor informations. So and this data, if they're matching very close or consistent with your data, then it is a very good model, right? Now, second is consistency of a model with the data can be measured by the probability that the data would be measured given the model that is being tested. Means consistency of a model with the data can be measured. Means your data should be consistent with the model. Suppose with slight side chain manipulation, your entire data of that model changes. That means that side chain is so important that it can change the main domain informations, right? So this is, you have to understand, your side chains will not change the main domain values. Okay, they are at the side chain, so their role is there, but that will not significantly change the entire value. If they are doing it, then your data is not robust and the consistency of your model is gone. Okay, so you have to see in different ways how robust is your data, to be very precise and simple. Okay, either you rotate it, you turn, twist, translate it, you take out one or two side chains and see the stability of your data, and if your data is still consistent with this particular model, then I think your likelihood, maximum likelihood increases, right? Again, coming to, yes, orientation and translation, which I already completed. So it is like a very straightforward, the use of likelihood in molecular replacement is straightforward. For translation search, using a model in a correct orientation. So a correct orientation will definitely increase the likelihood, but it should not altogether go bizarre and erratic, but your robustness can be checked. So your likelihood will increase there. And last, the methods are more robust and generally give clearer solutions in difficult cases that do Peterson-based MR methods. So likelihood methods compared to Peterson-based MR methods are a little bit more robust, because you are using not only a single model prediction tool, but you're using multiple model prediction tools, putting them together, coming up with a structure which is much more robust. The likelihood of them is very high, and the maximum likelihood this method will be more robust, and it will give a very clear solution in difficult cases compared to the Peterson based MR methods alone. Okay, so now improving on the experimental phases, that's what I was trying. So how you're improving? Now this is having a mean phase error of 53. So there is a 53-degree phase error, and this is the structure which we have got. So if you see this zigzag mesh type thing, this mesh is basically the electron cloud based on the intensity. Okay, so this intensity, what we have got is intensity of the diffracted X-ray light, which we collected in the form of a pattern, and this intensity is because of the X-ray passing through different electronic clouds. Now this electronic clouds is basically represented in this format. So what we are trying to do is, from the diffracted spot, we are getting the electron intensities, and this electron intensities in the form of a mesh, like a net. You can see like a net it is formed, and from the phase information we are getting this different angles and structures. So it is not a linear form, which is very obvious, and it is very crucial. So both it has to be, because the entire, angularity, the angle, the theta, phi, psi, all these angles are very, very important, and they give the final structure to the protein. And once the protein in its actual structure or form is present, then only its function can be predicted. Otherwise, what do you mean by denaturing of protein? It is simply when you have this structural protein and you apply temperature or some pH change or any buffer, and you break the bonds, and because of that it becomes a linear straight line structure. Then you can, at least, yes, you can find the sequence of amino acids, you can find what is the chain, what are the side chains, everything, but you are missing on the functional activity of that protein, because that protein is no more functional. And that information, if you are losing, then I think you are losing quite a bit of the information, because the functions of the protein are very interesting and they are something which you have to predict. Now, if we can improve this phase improvement, so if we see the phase error has been reduced from 53 degree to 41 degree, you see this loosely, mesh has become much more robust and tight, and this model, which almost looks similar to the previous model, but you see the orientation pattern, it was almost missing here. This have taken a firm shape, and this model is fitting, or this model has a better fit compared to the previous one. Okay, and that is how you keep on improving on your experimental phases by using the different methods and try out to get the best equation. Now, coming to electron density map. So this is the electron density map. You see this mesh, blue color mesh, these are basically electron densities. They are marked with different markers. So these are markers of different, it can be amino acids. So different amino acids are marked here for known reference. And what happens is, this is called chicken wire. Okay, so chicken wire, basically this mesh is known as chicken wire. And this is the computer understands the electron density map and gives you this chicken wire, and you find a structure like this. But knowing the primary sequence of a protein or nucleic acids, the atoms can be placed by hand into the electron density map. So primary sequence in a straight, like if you have this one, it is more or less like a straight linear form, you can do it, and it is a rough inaccurate model of protein structure. So just using the electron density map, okay, just using the electron density map, you can get a rough idea about your protein structure. You can get the more or less outline, but it will be a rough and inaccurate model of protein structure. So you need to have your phase information along with it. Now you have to do is, so this is just like from the previous one, you have all the informations, and once you have all the informations, what you have to see is robustness and refinement. Now what do I mean by refinement? Refinement is stability, basically the stability of your structure. So you've come up with a model of your native protein, and you believe that this particular model is robust enough, but you have to find it. So this robustness is, what you do is, the model is subtly changed. You do multiple iterations. Iterations means you do the same process again and again, and every time your model should look similarly to the present model. So every time after repeatedly doing it, what you are getting is, you are getting the same model. That means your model is robust enough. Every time you're getting the same thing. Then what you do is, you do the structure factors. So that is a structural factor F value, which is calculated, and you compare with the R of the crystal factor. That is the robustness of the, or R is the structure of the crystal. So you compare it with the R of the crystal factor. Okay, and then you see, or you with the find out with the measured value and how well this model fits the data. So for example, if I have to find out R of the crystal, that what is the, or in terms, you just take R as the robustness. Okay, what is the robustness of my structure? So what I know is, I have the structure factor of the calculated one from the model which I generated. I also have the F value from the native protein which I have done, that is the F observed. This is the observed one, this is the calculated one. And if I put this in this expression, so summation of all the observed mod of function of observed minus mod of function of calculated divided by summation of all the mod of function of observed, I will get the R value. And this value will tell me what is the stability or what is the robustness, how good is my model has been, how good structure, and ideally it should be more than 20%, 0.2, so it should be more than 20%. If it is if it is not 20%, then you have to think of it. Okay, so differences are minimalized, and as you can see here, 0.2, so basically it is in this range. If it is a good structure, it will be something of 0.2. R should come to 0.2. Okay, now there are also problems because these are all predictions, right? So this is the formula by which you can do it, but the problem is the R factor can be artificially lowered by adding more solvent. So when you add more solvent, these numbers change, and then R factor can be you can say altered or manipulated. Okay, that is one factor. Better R free as R crystal. Okay, so the ratio of R free versus R crystal, but calculated with 10% reflections which have not been used for refinement. So unbiased data set, because you have to also understand what is there. There may be free, there may be bounded atoms. So the free as well as bounded, that is R in the crystal, and calculated with 10% reflections which have not been used for refinement. So this has not been done for refinement, but we have to do it. We have to see that with 10% reflections, what is the biasness, unbiased data set I am able to achieve, and this unbiased data will tell something. So this is a problem. It is not that easy. You cannot do it as simply by using a 10%. So this is a problem where you have to be skeptical enough, but yes, taking care of it, we can think of the resolution part. Now resolution, yes, the quality of the crystal, whatever the quality of the crystal you're using, the conformation inhomogeneities, the mobilities of the individual atoms. So this is particular one structure. You see, this is 6 angstrom, this is 2 angstrom, 1.5 and 1.1 angstrom resolution. So as I'm improving on the resolution, what I'm getting, you see these are becoming much more sharper, and you see this is basically the electron cloud, that is the mesh, which you can see electron density. Okay, now with 5 angstrom you see a helices roughly visible. With 3 angstrom the peptide chain visibility increases. You have chains, side chains if sequence known. You can also see the side chains with 3 angstrom resolution. With 2 angstrom the conformations of side chains are visible. So as we lower down on the resolution, what is happening is your quality of the crystal and the conformations are becoming much more prominent, and you have a much better. Now, number of unique reflections used for structure determination, so at least 4 times more than the atoms required. The resolution should be, it should not be limited. It should not be. It is, if you say it is limited by, yes, these things are there, so you have to take care of these things. The unique reflections, it has to be almost like 4 times more than the atoms required. Okay, so that many angles of reflections or plane of unique reflections. Why I'm saying unique reflections? Because if you change the angle slightly, if you change the rotations, if you change the angle of the X-ray beam, then all these unique reflections you'll be able to get it. Okay, and typically this is like not by wavelength changing. So here it is entirely on the angular part. So it's not the wavelength change, it is the angle change, and getting the unique reflections in each of these angles and taking that data out of it. Okay, obviously temperature Temperature is also very important because it describes, the temperature actually helps you to give the local imprecisions and electron density. So it will give the information about the local imprecisions. There are changes in the electron density. It can be also like the temperature can also help you to calculate the structural refinement. So wherever there are slight impressions and all from the temperature factor, you can calculate that. So you have to calculate the B value. Okay, the B value will come here and it is measured in angstrom square. So a good B value or a good protein structure will have a B value of within the range of 20 to 30 angstrom square. Right? So this angstrom square is the unit, and now you have to understand higher the B factor, the more imprecise the atom position. So local imprecision, precision is precise. If it is not in the position, then it is called imprecision. The local imprecision is given by the B value. If the B value goes higher, the higher the B value, the more the imprecise the atom positions are. Okay, so what can be the reasons for this higher B factor? The thermal motion of the atoms. The thermal motion of the atoms, you see here, they can cause, because there are a lot of temperature changes, there are movements there which are increasing due to the temperatures. So thermal motions of atoms, they can contribute to the higher B factor. Different conformations of side chains, the lower occupancy at the specific position, higher occupancy at the particular position. So different conformations, the side chains are not fixed, their conformations keeps changing from one to the other, and the lower and the higher occupancy can also can be a reason to a higher conformation, higher B conformation. Now this is an important thing. So if the conformation or the higher occupancy, so you see here lower occupancy and specific position, what will happen is there will be a biasness. Because of this biasness, your B value will not be perfect. So your fluctuations will be there. And obviously the last but not the least is the protein disordered. So if a protein is disordered, that is in surface exposed loops. So if you have in surface exposed loops are there, and it is disordered in that case, because you see here just one loop in the surface of the protein, and that is in a lower occupancy. Okay, so that is not in a densely crowded electronic structure. Like if you see in this case, they are all having a pattern and they are in a position where they are distributed equally. Now in this case, suppose this is the actual structure, forget about this. In this, if this is one, suppose or I take one side chain, any of the side chain, and there you have just one loop coming out from the side chain. So what will happen, because of that the B value will be changed, and you will have to have a higher B value, which will give the protein disordered information. Okay, right. Now again coming back to the Ramachandran plot, because this plot will give you the idea about your structure and the protein backbones. And from this, what you have to do, you should have only allowed conformations. So the protein backbone should have only allowed the conformation. So conformations are there, and completeness of the structure should be more than 90%. So once your model is almost more than 90%, then only you can come. Now you see here, you can see the these are the alpha, beta sheet 2, and all this are distributed. You can see here psi, phi, this angles and degrees are distributed in the Ramachandran plot. So about the Ramachandran plot we discussed in the previous session. So you have to keep that in mind. And here, once you plot it, you find out what is the actual protein backbone which I've got. But this will only work when your protein structure is more than 90% complete. Otherwise, this will not give. So finally, when you get the data, Okay, so when you get the data, you see a table. I'll show you I'm showing you a table here. This is X-ray data collections and refinement statistics. Here the data collection, the space group cell, you see here A, B, C, alpha, beta, gamma, and based on this C2 and C2, these are two conformations, and in these two conformations, these are the various, like the completedness, multiplicity, resolution. Okay, a refinement has been done, and refinement with resolution. You have seen the resolution here. No reflections, work or free. So it's number of reflections, work or free. Then you have number of atoms. Okay, average of the B value here in Armstrong square. Okay, the receptors. So all these anisotropic parameters in terms of B, and then finally the Ramachandran plot. So all these informations, when you get it and finally it is ready, so you are ready to say my plot or my entire model is complete. Okay, so it is not only like XYZ, it's not only that just the angles, but you need to have all the complete spatial information of that crystal, and then only you can come up with it. Now for your like at the end of this session, you can go to this protein database. So PDB, it is RCSB protein data bank, and in this data bank you have lot of, there are other also like Rasmol, Swiss PDB viewer. So there are many you can say the protein data banks are there. You can find out, you can explore, and you'll see all they are given in this format. So structures, not only the structural data in XYZ coordinate of each atom are submitted to this, but freely there are also like other angular informations, B values, F values, phase values, everything will be given, and you can freely access all this. These are downloadable, you can find out, and there are a lot of softwares are also there, and in this software you try to get one and then see the structure. In that structure you make small changes. You know, remove one side chain, you add one side chain. Okay, you change the conformations, you change the rotate it at another angles, and see what are the what is happening with the model. Okay, so to summarize the entire thing, what we started with X-ray patterns, right? Diffracted patterns, and different patterns are there. We started indexing these patterns, from these patterns indexing, and indexing what in different planes, XYZ planes or HKL, and this HKL had together integrated for all this, and then found a proper strategy. Within this strategy, what model it will fit based on the multiple images, based on the Patterson expression, based on the other models like MR and other suitable model fitting. You got the phase information. Once you got the F value, once you got the phase value, you try to get it into a model, and then you can have multiple of them together, and once you get the multiple of the models together, you come up with a optimized structure, and try out with its like B values, try out with its resolution, and do various refinement. Once you have done that, you come up with the original plan of data collection. I will show it little bigger so that it is easy. So you see the original plan of data collection. Resolution is selected according to the user's choice, attenuations at 1, and so the resolution limit 2.2 angstrom, anomalous data phi, you see the phi from 0 to 360.45. Total rotation range is yes, this one. Number of images 267. Overall completedness, that is the structural completedness, is 97.3%, and you have a redundancy of 2%. Now the R factor of the outer shell is 3, as there is 27.1, and you have the the sigma factor is the outer shell. Again, the total exposure time, total data collection time. So you see here, in just 717 seconds, you are able to collect this huge information and then get the structure of it. Okay, so you see here all these informations with respect to how many 267 number of images. So these are 267 all data. I'm not showing it here, because of the constraint of the space. But this is how you collect your complete data and then give the structural information. So data processing, substructure determination, phase calculation, electron density maps, they all go hand in hand. Now you see here from here the you can see the first one, yes, say anomalous versus resolution, and this is, so I think the best one will be this one, the original and the inverted. So you do the contrast into multiple cycles, and after that you find out whether they are good fit or not, and this gives the electron density first one. Finally you will get the actual structure of your protein based on the previous all this informations. Okay, so I think it is like I've tried to give a very comprehensive idea without missing out on key aspects, how you can start with basic pure protein, get a crystal out of that pure protein. Once that crystal is there, why we are using a crystal, what is the purpose of having a crystal, that is also clear. Then we are using X-ray, why you are using an X-ray. Okay, so this X-ray will fall onto different parts of this crystal. We will talk of a unit cell, different planes of reflection, and then diffraction will take place. The diffracted, scattered X-ray images will be taken at different angles, and from multiple angles this data will be collected. Then from there the electron density will be calculated, then the function, structural function will be calculated. We have different expressions to calculate the structural functions. One, we have the electron density information map, then we have to find the phase information. There are different methods. From there we'll collect the phase information, put with the electron density informations, and then try to come up with models. Then there are different models. Once we try to fit with that model, the best model, and see the robustness of that model, change the angle, and then finally come up with the structure. Once we come up with the structure, we'll test the robustness, we'll test the B values, the resolution and refinement, and finally come up with the structural elucidation of that pure protein. Okay, so this is the entire process. These are some of the references. Shell X, basically this is the software which is very widely popularly used these days to find the structural elucidations of proteins. Okay, so with this I have come to the end. Any questions, please feel free to ask me.